MBA IM289V. So this is how you can reach me, either via email or you can always call my office. Happy to have you there, contacting me and so forth. And Dr. Frank Schwang here, who is also instructor, at least from my point of view, instructor of record. He's not an official professor in the university, so he has a hard time carrying that particular title, but he is the person who makes this course run, coordinates it, and so forth. So we'll be reviewing, basically, the field of biophotonics, what you can do with it, and so forth. And what I'd like to, if I can get my computer to change here. I'm Dennis Matthews. I'm the director of the Center for Biophotonics. I'm also a professor here in UC Davis in the Department of Applied Sciences and in BME and in neurological surgery. And primarily, I focus on this field of biophotonics. Hope to get you excited about it. It's kind of hard to lecture to such a few number of people and one that I already know, but I'll try to give it my best. So Dr. Frank Schwang here, who's an MD PhD, who helps me with this course. He coordinates it, lectures in it, is this other lecturers, and so forth. This is a team taught course, so I'll teach the first lecture, and I think maybe parts of the last lecture, and there'll be some very noteworthy people in between. It's a nice way for you to learn from the masters of the particular subject. We oftentimes have somebody lecturing even from afar. I don't know how many we have this time. So you'll actually get to hear somebody via televideo who is a worldwide expert in a specific area of interest. And I'll go through the course syllabus that we've prepared so far. So lectures I've just been talking about. And the grades are basically based on your attendance. I like to see you here. And even if I'm not here, I'll be monitoring via televideo. And there are some paper at the end of the course, term paper. And there are some problem assignments that we typically put out during the course of the lectures. The term paper is due to me within a week after the last lecture. I think this year that might be of order March 20th, but I need to check that date. But certainly before your final exams are over with. What I want in the paper is a summary of what you learned from each lecture, just to prove to me that you came. Don't turn that into a vast document. That's typically a few sentences, maybe a little paragraph from each lecture. And then more importantly, really, is tell me how you could apply biophotonics that you've learned in this course to your field of research that you're doing with whomever yet. And if you don't have a field of research, well, then let your imagination run wild about how you might apply it. I've had some very interesting lectures so far. Some very interesting papers so far come out of our classes. And we have a worldwide website, a web portal for our field called biophotonicsworld.org. And occasionally we ask students to contribute to that. What we have them contribute are either some write-ups on particular subjects we might be interested in. We have them do some book club type things, library club or journal club type things where you look at a certain journal article and give us your impression of what was the salient features of that journal article and what you thought might be wrong with it. It makes some interesting debate that we can put into this worldwide, if you like, forum for discussing the field of biophotonics. And we've been slowly building that over the years. The notes, PowerPoint lectures, actually I think they'll be in PDF, and the course agenda are available at this particular website. I will make sure you get a copy of each lecture. I think this one goes up on that website immediately afterwards. For now, if you just go to the CBST website, www.cbst.ucdavis.edu, and you go to courses and search down to this particular course by its title and I think by its course numbers, you can click right in there and go in and see the lectures. Each one will be there immediately after, and sometimes we even get them there before the lectures.
You'll also see course content from previous years. This is our fourth year.
Right.
Class. So
right.
Just make sure you click on the 2009 version, not 2008.
Right. And then the other thing is they are streamed. I believe it's free streamed And so if you see, if you click on that particular course, it will give you some instructions on how to view it on your own computer. So you can either see it real time on your own computer. I'd rather you were here, so it's probably not a good idea. But on the other hand, you can go back and replay the lectures if you wish to see something again and again, that it was something perhaps that you wanted to learn a little bit more about. And that's the whole idea behind the webcast and on-demand replay of the lectures. And as I mentioned already, some of your instructors will be teaching from remote locations. I don't know how many this time. I guess we'll show that in a minute. Two of this time. Okay, the people from Roswell Park Cancer Center. So did you want to add anything to this before I go to the actual lectures?
No, so I'll give, so there's going to be a list of lectures and topics. Every Tuesday, Thursday is going to be the course. It's a pretty full schedule this time. There are a couple of people who haven't confirmed yet, which we're working on. I may substitute in as one of the people who, so the schedule here is on real-time hyperspectral imaging. I may take that spot to cover clinical applications of that time. Right? But pretty much this is the list of topics. And then also just kind of as a way of giving you a heads up or thinking about what it is you want to try to get out of these talks. All right. By the end of this course, you should be able to answer these questions. And so I just kind of drafted this up.
Me too.
Yeah. Right. But
okay.
just to make sure that you do get something out of this course and not just kind of zone out listening. You know, as you go through these courses, you should, if you don't already know the answer to these questions, you should be able to answer it by the end of the course.
In other words, we consider this the principal takeaways from the course. Yeah.
Okay. And don't be surprised if maybe you see this again for the end of this class.
Right, so here's the syllabus. As we just talked about a little bit, I give the first talk, Sebastian Waxman-Hoju, who is our facility director at the Center for Biophotonics, will give the second one. Stefan Marchesini will talk about molecular imaging. It's a very high priority project of ours using free electron lasers, X-ray lasers. Steve Lane will talk about the overview of optical microscopy. He's an expert in that particular area. Thomas Huser will talk about nonlinear optical methods. Jack Rutledge, who you know, of course, will talk about optical methods in lipid and metabolism research. You're lucky here because oftentimes these professors have their students actually give Cynthia Pagba, who's one of our postdocs, will talk about surface enhanced Raman scattering as a method for sensing. James Chan will talk about CARS for sensing and imaging applications. Nathan, yeah, that's right. Rockwell will talk about.
What is the red and yellow?
So yellow means I haven't confirmed with them yet. So
right.
That's still kind of up to possible revisions. Roswell Park shouldn't be yellow, and then red is somebody I need to.
Yeah, Stavros Demos on real-time hyperspectral and multimodal endoscopy.
I'll talk to him.
Right. I was just talking to him today. I think he'll come around, but we'll see. So it's usually not a problem to fill up these slots. It's just there might be some sliding of the exact lecture, and you'll know that. We'll put it on the website. So anyway, I think we tried to give you a cross-section of the researchers in our own center and in a few instances from outside the center, like you say, the Roswell Park Cancer Institute. They're real experts in photodynamic therapy. We've had another guy who normally lectures there, Brian Wilson, but this time we thought we would substitute these folks. And Chris Contag, if he lectures, the postdoc that normally worked with him, Rajesh Shin, has now left, so we have to make sure we can get him, Chris, to actually give this particular lecture, and we'll know well in advance of when that will occur.
He did last year, so he spoke to his office.
Right. So, again, I think at the end of all this, you're going to understand quite a bit about the field of biophotonics, where are its new vistas, and what's really hot and happening in the area of our research at this point. So, what I want you to get out of this course, Frank talked about the takeaways. This is probably a little more generic. I really want you to, I want to introduce the field of biophotonics to you. What is it? How does it impact bioscience and medicine? I want to describe biophotonics markets to you. This is what I'm going to do in my lecture today. And industry players. The reason I want to talk to you about this is there's jobs for you and tools for your research needs that come out of that. I want to introduce you to researchers and research in the NSF Center for Biophotonics. I've already been talking about that in the syllabus here. And I want to tell you about some of the new challenges that could make interesting PhD projects. I don't know. He's now left the room and his cell phone's going crazy. And then I want to speculate a little bit with you about the future of biophotonics. I think it's got a very promising future. I'm going to have to move up here a little bit because I usually take prompting from my computer so I don't have to continuously look at the screen. So what is biophotonics? Let's make sure we're all on the same page. It's got several names. It's been called biooptics. It's been called biomedical optics. These, as far as I'm concerned, are all synonymous. So there are big meetings on biomedical optics. The same people who do biophotonics come to those meetings. We chose the word biophotonics because we just personally liked the ring of it. We thought the whole idea of applying photon-based technologies to medicine and bioscience was better, if you like, characterized by using that term. So we are a science and technology center, and that's why we talk about it in terms of not only what the science is of biophotonics. It's the science that investigates the fundamental interactions of photons with matter, and in particular biological matter. It's also, though, the technology that we develop in order to do that uses light to observe or manipulate biological systems. And clearly one manipulation of them is to do surgery, if we wanted to, even on a cell or on a whole organism, somebody's brain and so forth, using optical light or a laser. And we've coined this phrase, which we've used all along, shedding light on life, as a way to basically typify the fact, or characterize the fact that light's being used to better understand life or to improve the quality of life. So our definition of biophotonics is this application of photon-based science and technology to biology and medicine. Oftentimes, and this is where we might differ a little bit with biomedical optics or with biooptics, the people there tend to concentrate on the visible and the infrared, maybe in the sort of near UV regime. We really like photons of any color, any energy. We would take them all. We even have done some radio wave experiments in the past, but for practical reasons, we run in the sort of microwave regime or far infrared all the way out into the gamma ray or hard X-ray regime. No reason why not to use those photons. No reason why not to use them for biophotonics. So some examples of biophotonics, imaging tools are shown here. Everything from using fluorescence imaging to diagnose, or in this case, observe a tumor, a labeled tumor inside of a mouse. The development of luminescent particles, in this case, quantum dots, nanoparticles that can luminesce at different wavelengths, as a matter of fact, in inverse proportion to the size of those particles. The use of, again, fluorescence microscopy, but in real-time imaging at relatively high number of frames per second to understand, for example, transfer of viruses from one cell to another cell. This is, again, the whole idea behind biophotonics, the whole idea behind using light, if you'd like to study living organisms, is to concentrate on the living part of it. We really don't want to do electron microscopy. It's a very powerful tool, but it has to work necessarily with stained and very dead samples. We better use our technology, that of light, because it does not perturb matter very much at all, to study living organisms. It gives us much more information as a result. Sensors and assays are another area of biophotonics. Everything from using a pulse oximeter. If you've ever been in an emergency room, then probably the first thing they stick on your finger, first thing they stick on you, period, is a pulse oximeter. That's to make sure that you are not suffering from some sort of malady that's reduced the blood oxygen level. We also use biophotonics in the sensor regime to study and to design new drugs. This is, could be a gene study using an array of different genes. It could be a protein interaction array, whatever it is, and we can label these arrays, label them with fluorescent materials to give us different information about each and every well, if you like, little microcuvette inside of these arrays. Another area using, again, nanoparticles is to put a small particle inside of a living cell. This is a Chinese hamster ovary cell. You put a small particle in there, you interrogate that particle with light, you can functionalize that particle so that you can measure local biochemistry inside the cell. You can use, in this particular case, we use a laser as a tweezer, we call it, where we use the force, the momentum that a focused laser beam can produce to actually move this particle around within the cell. So it gives us a very powerful nanosensor technique inside of a living cell. Move it anywhere I want. I can move it to the cell wall, the cell membrane. I don't know what's wrong with this thing, frankly. Every time I press it, it stops. I can move it to the cell membrane or the cell wall. So if I want to measure, for example, if I had this inside of one of these little microcuvettes here with a certain drug interacting with the outside of the cell, I could see if that drug is making it through the cell membrane. I can see what it's doing to the local microorganisms inside of that region of the cell. I can move it over here next to the cell nucleus. Basically, I can use it to study anything I can functionalize it to do inside the cell, and do so non-invasively, other than I had to put the small thing in there to begin with. But the cells have a tendency to endocytose these particles anyway. And of course, in clinical diagnostics and therapy, and Frank can elaborate on this more, I think, in your lecture later in the course, we do everything from surgery with lasers. This is my co-director of the center, Jim Bogan, who is a neurosurgeon. He's actually performing surgery using the laser at this point. So basically, the laser is a cutting tool. It's a blading material. Yes?
Aren't most surgeries going to use that, right?
Not really. I wish they did, and we would like to make that happen, and we're working on some technologies to do. The reason they don't do it so often is, and with some exceptions, largely in cosmetic surgery is where it's used now. The reason has been that the laser, as it was originally brought to bear on surgery, was a very crude tool, and it would have, damaged far more tissue than I think it would have improved the outcomes for, because it was not the right wavelength of laser, it wasn't the right pulse duration of the laser, and so forth. So as a matter of fact, you wound up heating up large areas, and most surgeons shied away from that unless they had a specific application like denaturing tissue or something like that.
Friend of the vascular surgeon here said, whenever you see a scalpel, it says, that guy's old school. He's a good man, like his nurse was like, I didn't know what a scalpel is, I didn't see it. Everything he was using to cut the skin was some kind of heating. Maybe it wasn't a laser, maybe it's just some kind of
electrocautery.
Yeah, oftentimes it's RF ablation or electrocautery device. Yeah. Right, but those again are fairly brutal. I've worked with some companies before who do that kind of research and do that kind of technology, and they're ubiquitous. They're used in many, many different surgical procedures. But we'd like to carry it a step or two further and use a laser really as a precision scalpel. So it's if you like, it's the Star Wars phenomena of a laser saber, and I think we can do that. And we have some ideas that we've been exploring for a number of years now to be able to perform surgery at a very precise scale. Saw a cell in half if you wanted to. I think it's more of an issue also of having the laser be guided by something other than your hand. In other words, the laser can be so precise because you can focus it down and you can deliver energy in a very small volume, that it exceeds the capability of the surgeon's hands movement, the actual motor coordination of his hand. Furthermore, the surgeon doesn't know necessarily which type of tissue to cut. So that's where spectroscopy plays a role and imaging plays a role up here, that we can literally, actually I've got an example of it here, this is hyperspectral microscopy. This is the lecture we're hoping to get from Professor or Dr. Demos, but is can we use hyperspectral microscopy to identify literally at the cellular level abnormal tissue within normal tissue? Once you see that, and if you have something that can demagnify the movements of your hands, like, for example, a robotic delivery system, then you can perform some very, very precise surgery that's guided by the fact that you've identified abnormal tissue to begin with. So you're using spectroscopy of the tissue, what wavelengths are either reflected by that tissue or are fluoresced by that tissue to give you an identification of abnormal tissue. And it turns out that tissue that is abnormal does reflect differently. Every doctor knows this, and tissue that fluoresces in different wavelengths from normal tissue also turns out to be quite abnormal. It can be cancerous tissue. It can be, oftentimes, mostly it's got the biochemistry of the tissue is changed by having very high metabolic activity, which typically, for example, a cancerous lesion or even malignant cells will have compared to a normal cell. This is an example of another area, and this is where the Roswell Park people will talk. This is someone applying or performing what's called photodynamic therapy. So in this particular case, they've injected a patient who's not very visible here, but a patient with a photosensitizing drug. And this drug is supposed to be preferentially uptaken, if you like, into a tumor, because the tumor is very hungry. It's absorbing more and more nutrients than it would normally do so if it was just an aggregation of benign materials. And then once it's absorbed that, then you shine light on it, and lights of certain wavelengths activate, if you like, these photochemical reactions. And they're very aggressive at destroying, basically, the cell membrane. And the cell, in this case, the cells are cancerous cells. So it turns out now to be an FDA-approved technique for treating cancer, and has some obvious, very good applications in areas which are hard to operate on, your esophagus, some other places in your lungs, and so forth, which might be hard to get to with a scalpel, as we were talking about a little bit earlier. But this is just a snapshot of some of the things done with Biophotonics. Some more examples. These are my favorite colors of different. This comes from Invitrogen Corporation, who got bought out by somebody else recently. But
Molecular probes got bought out by...
Sorry, Molecular Probes got bought out. That's it, so my apologies. Anyway, these are showing labeling, again, fluorescent labeling of different parts of the cell. The nucleus of the cell can, of course, be labeled differently in color than the surrounding areas of the cell. This is same sort of thing you could see, if you like, in either stained dead material or in electron microscopy, but in this case, these cells can very much be in vitro. They can be living on a microscope slide and so forth, which gives us extra capabilities. Again, just various different types of cells. This is even a contrast microscopy version of it. But in addition to labeling cells either by whatever they have endogenously contained within the cell, which sometimes can fluoresce at different wavelengths than the light that we shine on them. But also we can look at time-dependent behavior. And this is some work from Paul French, where he does microscopy, but and Professor Mark Kuh here in bioengineering does this similar work. But this is an image where we're looking at endogenous labels. So basically chromophores within the cell that could fluoresce. But we're looking at those that have different lifetimes. So we send a short pulse instead of a CW form of light. We send in a short pulse of light, and then we look at how the fluorescent light decays in time. So, and from that lifetime, which usually has an exponential component to it, to the decay of intensity, we can determine different materials, the location of different materials. And the advantage of this technique is that I don't have to stain it with any kind of external chromophore. It's all done with internal chromophore. You can add some if you want, but you don't necessarily have to. It's a very powerful technique. And it uses a short pulse laser to initiate, if you like, the fluorescent cascade that gives us different or exponential decay to the intensity versus time. I keep looking at that clock, who's completely off. One of my favorite parts of biophotonics, so again, I like looking at living things, is shown here. This is optical coherence tomography image done by Joseph Ivesat and his colleagues and published in the Journal of Optics, I think, a few years back. But what this shows is a phenomenon, this is also an example of it, but it's not moving, where you scatter light at different depths off of different materials. And if you have interfaces, the light has a tendency to scatter even more. And what you're seeing here is a zebrafish, a living zebrafish. And this is exactly the image you would see if you did histopathology. You cut a slice of this, or took a little plug of tissue, if you like, out of this fish, killed it probably, or you could in the process, took it away, stained it, and put it under a microscope. But now we're able to do this for a living animal. Just because we can look with very high precision, we can do this at microscopic resolutions of a few microns, even less under certain instances. And we can obviously do it real time, because you can see this fish, his heart is beating inside of the skin. So this is the outer skin, this is some inner material behind that skin of the fish, and this is the heart of the fish beating, obviously slowed down because it beats very, very fast. Another example is something we've done with a company that was called Inframed, called ViewStick now, I believe, where we take advantage of the fact that light that scatters off of tissue, infrared light in particular, scatters off of tissue, will scatter with different intensity off of connective tissue versus vascular tissue. And so, in this instance, they have used this as a means to identify or locate veins in someone's arm. And if you're like me, I don't like to be poked more than 2 times. Third time, I usually start to get a little woozy. I was lucky there the other day when we were in hospital. They got me right away. But anyway, this is a way, and this company I think is going to do well with this technique, to identify for the phlebotomist where are the vessels of interest and use that to extract blood from. Yeah.
But the real application for OCT, the penetration depth is very low. So would that actually be a real application on human-based objects?
Yeah, I mean, I've got an example of one here from Dr. Brzezinski in, I think he's still at MIT. He was certainly working with Jim Fujimoto there, Jim Fujimoto being a pioneer of optical coherence tomography. And this is showing a cutaway section, but it's not really cutaway. It's got a catheter going down it of a vessel, somebody's blood vessel. In this case, it's an artery. And what you can see, and the way they facilitate this is this was probably a dead vessel, to be honest about this one, but they can do this now with living tissue, and they flush the region with water when they take the image. And the OCT, or the wavelength of light they're using for OCT, penetrates water pretty well. It'll go, centimeters in that, and then scatters off the vessel wall. And of course, what they're trying to identify here is this plaque, calcific plaque, that's located next to the intima of this artery. So there's some very nice work coming out on that. And again, they take advantage of the fact they can only go, or they get around the problem of it only being able to go a few millimeters, probably in practice a couple of millimeters in tissue, by flushing the other area that it has to go through, the other volume it has to go through with water, so they don't have to get it all absorbed in, for example, the blood inside the artery. So I think there's a great application for OCT. It's probably going to be one of the most important contributions in the recent term. And I think it's for all kinds of endoscopy. If you want to look at delamination inside your colon, you want to look at your teeth to see if you can find caries in them, and so forth. It's going to be pretty powerful for doing it.
Do you think it's also possible to implement Raman into that?
Yeah, that's a good idea. I think there's several researchers I know around the country, and since I'm being recorded, I'm not going to say their names right now because I might get them wrong. But there are people who are considering doing that, at least several different institutions I know of.
That would be something really big.
I agree with you. So if you do Raman, you can even talk about doing fluorescence. You can talk about doing polarization-sensitive scattering. There's all kinds of things you can do with OCT. But again, you're taking OCT functions that you don't already know by. It's an interferometric process. They can choose at what depth they look at the scattering by adjusting an interferometer. And you can do that some fancier ways with the Fourier spectral analysis and so forth. But that's the general principle. Look at choose a certain depth. You want to look at the scattering power of the object that you're shooting light into.
And spectral analysis.
You could do the same.
Yeah, no, I mean, like, if you would also, like, have, like, this from
You could, yeah, I mean, there's no reason you couldn't do hyperspectral imaging at the same time, or hyperspectral OCT, if you like, at the same time you're just looking at the scattering power. Because this is monospectral.
Mm-hmm. Yeah.
You know, probably seen this slide, Yvonne, but this is my favorite slide of what the power of OCT. This is showing a bunch of different cells, if you like, laid out on a grid, and all of them we're able to observe at once. These are in fluorescence, and we're doing this with a spinning disc confocal microscope. But that gives us the capability, if you like, to see cells, a lot of different cells, not having to study just one cell and see what happens, but to see many different cells. In this particular case, there's one cell, a T cell, that's been labeled with a protein, that's been infected with HIV, and in the GAG protein of the HIV, there is a green fluorescent protein label. And so we can see that fluorescence, and we can see when this protein associated with the HIV infection at a point, this is a little area where the two cells are coming into contact with one another, an infected cell with a non-infected cell. We can see that the infection, in some instances here, I got to find one. Here's one where the infection is starting to spread over. You see a new nucleation group here of proteins that have moved over into another cell. Believe it or not, there is a new cell here. It can't be seen because it isn't fluorescent yet. But this cell has come into contact with it, and we're very interested in this region. It's called the synapse region between the two cells, and there are protein interactions taking place in that synapse region that enable this cell to infect the parent cell to infect the daughter cell or the host cell. A lot of research institutions around the world. I just bring some of them to your attention. I haven't covered anywhere near all of them. That's the purpose of our Biophotonics world. But not only us, of course, but many, many other places. The Britain Chance Center I visited this year, so it's Dr. Schwang back there. In China, there's a great group in Queensland, the Russian groups, and so forth. So the reason I bring this to your attention is that if you work in our field, there's also a lot of other places you could work later on, not just even in the United States, but even in different groups around the world. But certainly there are other groups in the United States. The Beckman Laser Institute, I'll be going down there later this week, is one of the primary researchers in the field. Biophotonicsworld.org, I mentioned already. It's a web portal for our field. It's really to provide information on who's doing what and where. I urge you to go look there, and you may be actually, as part of this course, contributing some information for that web portal. Talks about jobs in our field, upcoming events, and so forth. We're actually putting together an initiative right now in Biophotonics for Life, which will be a consortium of probably 12 institutions around the world, some of which I showed on the previous view graph, but that will be engaged in developing biophotonics for their region, and will use partner institutions, if you like, from other places in the world who have expertise that they don't necessarily have. So it's a growing field, one that has a lot of interest from other places. And speaking of that, I wanted to talk about Biophotonics Marketplace. We did a study a few years back, 2007, which came up with a number like $53 billion a year for the worldwide market. There's a lot of non-medical applications like biometrics, scanning your fingers to determine your identity, scanning your retina, and so forth. That's based on biophotonics. But there's also tests and components, development of probes, which are these fluorescent agents that you can label different parts of the cell that I've shown you already. Put stars on things that I think are going to really grow a lot in the future. Medical therapeutics and medical diagnostics. This whole area is obviously got some big market shares here, 8 to 30 billion. And many of these are growing. That's interesting. Oh yeah, 30 billion, right? I read that wrong. I thought it was 37 million. This whole area is what we call theranostics, therapeutics or diagnostics. Somewhere between 8 and 30% growth rate in these areas. There's been several other market studies that have come out since this one. Pretty much all agree. Actually, some of them are even more aggressive, I would say, in terms of what the market size is and how big it's going to be. One of them I saw showed that by 2020, I think it was, this would be like 190 billion. A lot of companies. We have a database on all these different companies, some of which you can see here. There's many, many more. Come see us if you'd like to learn what products they sell and so forth. And Biophotonics is sort of broken down into, as again, I've already been talking about imaging and microscopy, sensors and assays and medical diagnostics and therapies. Some of these get pretty wacky. I like to sometimes talk about things like low-level laser therapy for regrowth of hair. So this is an FDA-approved diagnostics for, or therapeutics, for hair loss. Yeah, I haven't tried it yet, but nevertheless, it made it through FDA testing, so it must have some promise. I can conjecture that it might be taking advantage of light, especially the warmth from light, in this case infrared light, could increase local perfusion, which could improve the viability of your hair cells, the follicles of your hair, and so forth. But there is some other studies coming out where, and we have some collaborators in this area, that people are trying to understand better how light interacts with nerves and how light can actually modulate nerves, either to reduce pain and or improve, again, sensitivity and or call for more perfusion. So the perfusion probably is going to improve the hair growth. This could also be wacky. I don't know. But certainly getting through the FDA with no small fee. So some current developments in biophotonics. We'll wrap up with that. Our center, which Yvonne certainly knows about. He's working over there, but Colin, maybe you haven't been to visit with us yet. We're located in Sacramento. We're very much about interdisciplinary, multidisciplinary research. We have throughout our center, which includes many more institutions than just UC Davis, we have about 120 PhD and MD researchers, and we typically have about 30 projects per year, and our budget right now is about $7 million. These are our partners. UC Davis is the lead institution, but we also have researchers from UC San Francisco, UT San Antonio, Alabama A&M, Lawrence Livermore Labs, for example, National Lab, UC Berkeley, and so forth. And there's actually, I don't show it here, but we have about 20 or so other institutions now, what we call affiliated institutions that work with us from around the world. Our projects are again broken down into this category of imaging, advanced imaging, molecular and cellular biophotonics, and medical biophotonics. What you're going to learn about in this course are all of these different technologies, because you're going to hear about it from the people who've actually pioneered it. So I don't want to spend a lot of time on it today, other than to say we are developing microscopes that work in resolutions and at the spatial resolution scale that you couldn't heretofore buy. Now we have commercialized them with a partner, and we're working on developing X-ray diffraction imaging, which will enable us to finally see the structure of a single biomolecule. So that's kind of a highlight of that. We developed new probes based upon phytochromes instead of fluorescent proteins. And in medicine, it's kind of a potpourri of where we see medical needs. Everything from developing new catheters that can treat people with atrial fibrillation, develop new means of deploying materials to seal off arteries or aneurysms in arteries, and we use light to do that. to optical disease detection. This is part of our work for the whole world of warfare, threat detection, point-of-care testing diagnostics. We just started a new center I'll mention here in a minute. So technology for biology, I kind of already hinted at this, is, this is my favorite cartoon about this particular thing. But I mean, how can we study the viability of a cell and do so, of course, without harming that cell? And so obviously it's light that we are interested in doing that with. And our whole goal here is to equal the resolution that you can get with electron microscope. But as I already talked about with you, I want to do this on on living samples. So I need to do it with some techniques, saturated structural illumination microscopy is one of those techniques that gets us down into this regime. You can see what kind of organisms that can be imaged with that kind of resolution. And then X-ray diffraction imaging puts us all the way down into the single atom and certainly the biomolecule structure determination regime. So can we equal that resolution? That's the key. That's what we've been working on in many of our research projects. And really, I think why is shown here. I was fascinated to find this particular image recently. It won one of these awards in Science Magazine and from the National Science Foundation. But it illustrates the difficulty we have to deal with. And every time I think we're doing a great job of imaging, this is where I get my comeuppance very quickly. And here, the challenge is really, and this is for a neuron, for example, is to have nanometer resolution so I can see these fine little features here. And this is an artist's conception, mind you, so I don't know what it really looks like, but from good biological knowledge guidance. But we have to have nanometer resolution. Oops, that's telling me I'm to shut up. We have to have nanometer resolution over the scale lengths of millimeters. These are millimeters in dimensions, and these are nanometers in dimensions. And we need to do it in 3 dimensions because there's a lot of 3-dimensional structure. And we need a time resolve because as, for example, calcium ions flow through a neuron, they change the voltage potentials in different areas of the neuron and so forth. So some of our neurobiologist colleagues have said, you do this, and our world becomes, we can go investigate the Holy Grail. We haven't gotten that far yet, but that's, I think, one of our biggest motivators to see if we can. And I actually think most of this is possible with, of course, continued development of the technology that we have at hand.
Yeah, I think they're working at the moment on metamaterials which have negative index of refraction, and they're kind of already brought a proposal for lenses which could
negative permittivity lenses,
it could basically bend the light beyond the resolution.
Right. You're no longer constrained by the normal materials.
That would be actually already kind of
it's possible. It's definitely possible. I think the biggest issue for this is going to be getting away from using, and that particular technology might get you there, but getting away from having to use fluorescent probes. Because once you start having to pinpoint all of this using fluorescence, it becomes a lot more cumbersome. It interferes with the functioning of the neuron and so forth. So can we do it with endogenous agents of one form or another? We got to have contrast, but how do we get that contrast? Can we look for Raman? Can we look for other
nonlinear
microscopy? But again, I got to have some kind of a contrast agent, right? Something's got to provide me with contrast, because if I don't see the difference between the scattering here and scattering there, I could have infinite resolution and I still wouldn't be able to identify things. So that's the key, is having contrast and having high resolution and being time resolved and being charge sensitive if you can for the neuron. But backing off a little bit, can I do something a little bit simpler, but not that much more? And that's what I was talking about earlier. Can we look at cells, one cell that's infecting another cell in that synapse region that I showed you earlier when I was talking about the fluorescent? Remember, I had that array of all these different cells interacting with one another. I'm very keen on what goes on in this synapse region. The virologists have said they need to better understand it. This is an electron micrograph, so of course it's not living. They can't see how all of this is changing in time. And as a matter of fact, Dr. Schwang is working with some colleagues of his in New York to better understand this. But I think this is possible because if we can use some of the techniques that you've been talking about, can we get down around 50 nanometer resolution, 20 nanometer resolution? That's about the size of these little buds, if you like, on the surface here. And this is a cell membrane being deformed by proteins that are on the surface. And once this is deformed, they also deform the host cell, the target cell, and then after that, you can get an injection of proteins that are associated with, in this case, GAG proteins associated with HIV. But the virologist has said, please go understand this for me. Please help me understand it. Help me image this in real time. So, we've been trying to do that starting with just real-time confocal spinning disk confocal microscopy. This again is showing that cell, the synapse region, and you'll see material start to spew over into the cell you can't see. This is another view of it where you have a contrast image, and therefore you can see the cell is not yet fluorescent. Fluorescent cell here, non-fluorescent cell there. Material spewing over. So we've got to improve our resolution of the synapse region to do what I was just talking about. And one of the ways we're trying to do that is with the OMX device, Optical Microscope Experimental, which we've now installed and we've commercialized with Applied Precision. The one that we've bought that is marketed commercially has about 2.5 times the diffraction limit. That's not high enough resolution yet to understand that synapse region, to visualize it. But we have another system that goes down to about 50 nanometer resolution, and that's starting to get there. So if we can put fluorescent tags on the synapse region and we can observe things at the 50 nanometer or less regime, and we can do so at 10 or 15 frames per second, I think we've got a chance of doing it. That's still not the neuron problem we talked about earlier, but it's getting there. So imagine that in 5 years we might be able to actually do the neuron problem with this and other nonlinear microscopy techniques that you hinted. But I think doing atomic scale imaging is going to take X-ray diffraction because we're talking, nanometer scale lengths here for the individual atoms in the biomolecules. And that's where we get into the X-ray free electron laser, new technology that puts out femtosecond photons in pulse duration, 10 to the 13 photons per pulse. They're fully coherent, which means they arrive at the same time and have some spatial relevance to one another, or relation to one another, and they're angstrom wavelength. These aren't built yet, but there's some in Germany, and there's one coming online here this year, actually, at the LINAC Coherent Light Source in Stanford. And the idea there is to send a short pulse of X-rays through a biomolecule and look at the diffraction pattern behind that. So the light, if you like, the lens in this case is the material you're imaging because the light that goes through it is scattered at different angles depending upon the electron density, if you like, locally inside that object. You get a very noisy pattern, but if you add up a bunch of these from continuing to put a stream in front of the free electron laser beam, you can reconstruct, and we think, at least at this point, get the actual structural information about a given biomolecule. And without having to crystallize it, which is the way it's done today. Protein crystallography with X-rays has been around a long time, but it's very tedious and cannot be done with all biomolecules. And a lot of those biomolecules are very important, like glucose.
That basically shows the angstrom distribution in the molecule?
That's what's doing the scattering. You can also do nuclear scattering if you want, but it's more abundant. It's a higher cross-section for scattering the X-rays off the electron cloud in the atoms. But that's fine. If you're scattering them off, if you can do things at that level of spatial dimensions, then you're going to image where they are. So I probably better shut down now. We do a lot of work on Raman spectroscopy, which enables us to look at the molecular footprint of cells by their spectral characteristics, vibrational spectral characteristics. I'll have to come back to that at a later lecture. You'll hear more about it. The future biophotonics I really wanted to end up on in this lecture, which is, how can we make medicine a little more appealing than going in like we do today for various ways of poking and prodding you and sucking blood out of you. So we think that biophotonics can help us get there. There's a lot of reasons to do that, but certainly one of them is we want to be able to diagnose you before you ever get sick, or certainly diagnose you very early into your sickness before you have so much quality of life decreasing as a function of long-time chronic ailments. And one of the things I wanted to tell you about, just in ending here, was this whole idea of doing prevention of disease by determining something about your genotype to begin with. So Affymetrix makes these gene chip systems. They look for, this is biophotonics based, by the way. You're looking at probably this is real-time PCR, polymerase chain reaction, but the real-time is looking for fluorescence of these SNPs that are associated with certain genes that have been shown to be correlated with disease, although that is somewhat contentious at this point. The whole idea here of what you eat and your lifestyle determining your health. I love this formula that your phenotype, who you are, is who your parents were and what they bestowed upon you, and the environment that you bring about for yourself. This lady says, I haven't got the genes, so it's okay for me to smoke. And this one says, I've got the genes, so I'm going to smoke anyway, since I'm doomed anyway.
Bruce German likes to add, plus the integral of birth over time.
Yeah, exactly. I like that. This is my favorite foods, which of course are not helping me at all. But as you said, nature may have loaded the gun from your parents here, but we pull the trigger with what we do to ourselves. So from a technology perspective, what's being done? For example, there's a company called 23andMe that has Oprah talks now about spit clubs or spit parties. People come and they spit in a vial. You send it to this company called 23andMe. They actually make a little kit for this, and they go off and determine your genotype. And then you're supposed to do something about it. There's another company I've read about, Genova Diagnostics, who does something similar, but then they have a whole host of supplements and physical trainers and so forth who can go presumably help you from succumbing to one of those things that you've been genetically predisposed to have. Very controversial here. I don't want to paint the wrong picture about how important this is, how accurate it is, but certainly the advice they give you and the supplements they give you are probably good for anyone, even if your genotype has got a A+, a good grade of health. But from a technology perspective, this particular technology here is Raman, which we're going to talk more about later on in the course. This is based on Raman scattering the light off of vibrational bonds in your skin. In this case, it's carotenoid antioxidant levels in the skin that it measures. And then this is linked to, again, what kind of dietary supplements you should have and so forth. This is another very small system I'm very interested in for point-of-care diagnostic, which is basically able to do a full-blown blood analysis of blood analytes and so forth. Again, it's based on fluorescence and real-time PCR. So last thing I'll say is we've put together a point-of-care testing center inside of CBEST and got some funding for it from the NIDIB. This one in particular is focused on, it's directed by Jerry Cost here, who's a pathologist, focused on pandemic medicine. But the idea is, can you develop technologies based upon, in this case, immunofluoroassay. There's many other technologies too. Some of them are shown here that can be used in the field to determine if someone has an infection, has been infected by a bioagent, or has Mother Nature infected them because of a virus or something that's been present. Even in this particular case, one of our colleagues has modified an early pregnancy detector looking for biomarkers that are associated with exposure to ionizing radiation. So if you came near a dirty bomb and you got exposed, could they determine post facto, because they didn't have a dosimeter on you at the time, but could they look at the biomarker levels in your blood and determine if you've been exposed to ionizing radiation? So a whole host of things that are being developed here, and every one of them is based on biophotonics. And so in closing then, my favorite prognostics here are, one, using your cell phone as a telediagnostic. We're trying to do what we call rural medicine. Got a whole initiative that we're putting together here, but this is this whole idea of using either the Bluetooth input to your cell phone. In this case, there's a company that's put together something called a glucophone. So in this case, they're taking a piece of, or a lancet or a micro lancet and getting a drop of your blood, looking at enzymatic reaction, looking at the color of a strip, reporting that out as a certain glucose level in your blood. We've been asked, and I'm sure others have too, to develop a non-invasive way of doing this because people don't like to have their fingers stuck. But I think it's interesting to see that the cell phone is becoming a platform, if you like, to report out this information to your healthcare provider or to other people who might be interested in this information. Also, this whole idea, and our Citrus organization here and at Berkeley is developing this technique of a cell phone as a telemicroscope. So this is the whole idea of having telepathology based on some person in the field in the jungles of Africa or whatever, who is, in this particular case, they're studying blood samples to see if someone has contracted malaria. Instead of having to send the blood sample back to have it analyzed, they take a picture of it on a microscope slide and send that to a pathologist who should be able instantaneously to say that's malaria or that's not. So again, telemicroscope based upon a cell phone or a glucometer, and you can extrapolate this to more and more other things that might be of interest. Zinc protoporphyrin levels to look for iron deficiency anemia, something that Dr. Schwann's working on. Also looking at the complete blood count for patients who are too sick to move. They've been having, for example, chemotherapy, and if you get their white blood cell count too low, they become incredibly susceptible to infection. So you have to then adjust how much chemotherapy that you give them. All these are people who need to have their some blood work done on, for example, or some microscopy done on, or pathology done on without having to move them, and that's what we're looking into. And that'll all be based upon biophotonics techniques. This is the reason we put together an Institute for Rural Medicine. This is starting between UC Davis and Tahoe Forest Hospital in Truckee. I won't plow you through all these, but needless to say, it's coming up with technologies that can be used to give people who live far from a hospital, for example, the same quality of health care as those who live right next door to the Mayo Clinic. So, that was all I really wanted to say in your first lecture. Give you a little appetizer, if you like, about what biophotonics is about and what it can do, and some ideas, at least on my own part, about what the future holds for it. But I look forward to your ideas and your learning about the field and your contributing to the field. Thank you.